Hello students, now this is the next video of the chapter Excretory System and in continuation with the chapter Life Processes, chapter 1 class 10th. Now in this chapter we will learn about the difference between excretion and osmoregulation, the different parts of the human excretory system and the physiology of human excretion. Now what do you understand by the term excretion? Excretion is the process by which we remove the unwanted waste from our body, or our body gets rid of the undigested waste or unwanted waste from the body. The same type of phenomena is called as osmoregulation. Osmoregulation is the process that maintains the amount of water and proper ionic balance. So excretion and osmoregulation are interrelated. One maintains the ionic balance of the body as well as water balance of the body, and excretion eliminates out the entire toxic waste from the body. Now what are the different functions of excretion? Its function is to remove the unwanted nitrogenous bases or nitrogenous compounds or nitrogenous waste from the body. Second is to remove the toxic chemicals that our body forms after the metabolism of the different drugs. It also maintains the ionic concentration or balance in the body fluids and regulates the pH of the body fluid. So if somebody asks you what are the different functions of excretion, so the first function is removal of undigested nitrogenous waste. Second is removal of toxic chemicals. Third is maintenance of proper ionic balance and osmotic concentration. And fourth is regulation of pH of the body fluids. Now there are different byproducts, and accordingly we say there are different modes of excretion. The first one is called as ammonotelic. Ammonotelic are those organisms which removes of ammonia from their body. Now ammonia excretion requires tremendous amount of water or large quantities of water. So this is specifically for the organisms which live underwater. So we can say aquatic organisms or aquatic animals are ammonotelic. The second one is ureotelic. Ureo means which excretes out urea. Now we are the ones which excrete out urea. The amount of water required in excretion of urea is comparatively less as that required in ammonotelic. So humans are ureotelic. The third one is uricotelic. Uricotelic is the excretion of uric acid. Uric acid requires very less amount of water. Birds are uricotelic, that is their excreta comprises of uric acid. Now we have different types of excretory organs in different organisms. Same way we also have a well-defined excretory system in our own body. So the human excretory system comprises of two major parts, that is the two kidneys. These kidneys are located at the back side of the body along the two sides of the vertebral column. The kidneys are bean-shaped brownish red structures and which range from a size to 12 centimeters. These kidneys are supplied with the blood vessels. The blood vessels are the renal arteries and the renal veins. The pure blood comes in from the aorta through the renal artery, and the impure blood goes to the vena cava through the renal vein. Now these kidneys comprises of the internal structure that we'll study in detail. After the kidneys there are two pipe-like structures which carry on the formed urine from the kidney into the ureters, then into the urinary bladder, which is a muscular structure which is responsible for the collection of urine, and then the urine is periodically ejected out of the body through an organ called as urethra. So four major parts of the human excretory system are kidneys, ureters, urinary bladder and urethra. Now we have seen kidney. The enlarged view of kidney and the internal structure of kidney is the bean-shaped structure is covered by a capsule which we call as a renal capsule. Kidney has two major portions. One is called as a renal cortex and renal medulla. So two major parts. If I cut a kidney and I could locate two major parts, one is called as the renal cortex, other is called as renal medulla. Here the structural and functional unit of kidney is present, which we call as nephron. Now I can say kidney is the filtration unit. Kidney filters blood and removes of the toxic waste. So how is that blood filtered? That is through the structures called as nephrons. So I can say nephron is the structural and functional unit of kidney. Then these nephrons are present both in the renal cortex area as well as in renal medulla. The nephrons that are present in renal cortex are called as cortical nephrons, and the ones that are present in renal medulla are called as juxtamedullary nephrons. These nephrons contain a collecting duct. The collection of all the ducts forms the pyramid-like structures called as the medullary pyramids, which open into a broad space. This is called as the pelvis, renal pelvis, and then it is transferred to the ducts called as ureters. They are innervated by a renal artery and a renal vein that brings in pure blood, impure blood, and takes away the pure blood from the kidney after being filtered of the entire nitrogenous waste as well as toxic chemicals and reabsorption of useful nutrients and electrolytes from the body fluids. Later on, we'll study the physiology of the human excretion by the structure of nephron. Now we start with physiology of urine formation. In physiology of urine formation, we will learn about the structure of nephron and we will learn about how the urine formation takes place with the help of these nephrons. In the previous topic, we have learned the structure of kidneys. We have learned that nephron is the structural and functional unit of kidney. Nephron comprises of the following parts. First is called as the glomerulus. The second is Bowman's capsule. Third, proximal convoluted tubule or PCT. Then comes the loop of Henle. Then it is further divided to form distal convoluted tubule, and then comes the collecting duct. Now these portions, here the glomerulus is basically the tuft of capillaries or a capillary network. These capillary networks are bringing the blood from the body or into the kidney to be filtered. Now this contains all waste materials, glucose, sodium ions, potassium ions, ammonium ions. All these gets filtered into this cup-shaped structure called as Bowman's capsule. Now how does the things get transferred? It is bringing all the substances. This Bowman's capsule is selectively permeable. They have a smaller pore size. Now whatever the substances that are reaching into the Bowman's capsule should be smaller than the pore size, then only it could be filtered. So this is a selectively permeable membrane which allows certain materials to be filtered, but along with it certain useful materials also gets filtered along with the waste. This process is referred to as ultrafiltration. Filtration means to filter out the substances. Ultra means at a very minute process. But the minute processes, the entire things is being filtered. So the things get filtered from where? Glomerulus to Bowman's capsule. Now this is referred to as the filtrate. This Bowman's capsule then meets the tubulated structure called as proximal convoluted tubule. In proximal convoluted tubule, whatever substances that have come into the filtrate, now because it contains some useful material also, these useful materials get reabsorbed. What is getting reabsorbed now? We need glucose, we need sodium ions, we need potassium ions. So these ions get filtered back into the capillary network. Now from here, this selective reabsorption is the second process in urine formation. The first one was ultrafiltration taking place between glomerulus and Bowman's capsule. The second process is selective reabsorption. In selective reabsorption, it is taking place in proximal convoluted tubule or PCT. Now the things that filtrate is coming into the loop of Henle. Here only the water gets absorbed, nothing else is getting absorbed from the loop of Henle. Now this filtrate gets transferred to distal convoluted tubule. In distal convoluted tubule,
many things that are still left in the capillary network gets actively secreted into this filtrate. So the third step is tubular secretion. We are secreting the things back into the DCT. So it is referred to as tubular secretion. So the things now that gets secreted into the filtrate would be traveled into the collecting duct. The last step in the urine formation is the concentration of this filtrate. The things get filtered, the filtrate then reabsorbs the water, excess water gets reabsorbed. We are drinking so much of water full day, but we are not excreting out the entire water. Body needs water, so we need to reabsorb this water. So now the water gets reabsorbed, the filtrate that was initially dilute is now concentrated. This concentrated structure or concentrated filtrate is now referred to as urine. This would be then transferred to the medullary. All the collecting ducts would combine to form the medullary pyramids. These medullary pyramids would be then transferred to the ureters. From the ureters they would collect into the urinary bladder. Once the urinary bladder gets filled 3/4, then the signals for evacuation of that urinary bladder takes place, and then urine is excreted out periodically via urethra. So the entire urine processes involve 3 steps: ultrafiltration, selective reabsorption, and tubular secretion. And the structure of nephron comprises of glomerulus, Bowman's capsule, proximal convoluted tubule, then loop of Henle, then distal convoluted tubule, and then collecting duct. So this is the entire structure of nephron and physiology of urine formation.